Welcome to the Student Faculty Seminar of the President's Lecture Series and the Philosophy Forum. It's my pleasure this afternoon to introduce Professor Colville. When I read her ETA, I was uncertain whether I should be depressed or inspired. Her accomplishments are daunting. More than a dozen books, 700 articles, very influential positions in government, on boards, organizations. And after I had worked my way through the 3 stages of depression and the 9 stages of envy, I did come away inspired. And so will you this evening when you get a detailed account of her accomplishments. Professor Colville will lecture in the University Theater at 8 o'clock on oceans, climate, and human health, the cholera paradigm. This afternoon, her topic is infectious diseases, climate, and the environment. Please join me in welcoming Professor Colville.
Thank you very much. I'm delighted to have the opportunity to speak to you. I hope my voice carries, does it? Good. I'm going to talk mostly about the environment this afternoon and about the marine environment and microbial species interactions in the environment. And this evening, that will be sort of laying the groundwork. And this evening, I will go into detail about several infectious diseases that are quite strongly related to the environment. So let me first start out by emphasizing that it's the interdisciplinary interactions that are the richest source of information in the 21st century. It's the interactions at the interfaces of science and engineering, the interface of the different disciplines that most of the discoveries are developing. And it's very much like the oceans where the polynyas occur and upwelling occurs, and that you have then nutrients that become abundant in organisms, populations develop. I'm going to speak about the marine environment particularly, and if you were to take a single sample of seawater, you'll find that it is comprised of large populations of microorganisms, very few of which we have been able to culture. I think the statistic is still holding of about 1% of the species that are present in a given teaspoon of water are cultured and described. Now, describing a species offers its own challenge. With the advent of DNA sequencing, genomics, and the concurrent metabolomics, proteomics that are ancillary to the understanding of a sequence, is understanding the difference between genera, and we think of it, at least I like to think of it as essentially a sieve, the differences that define species and the differences between isolates that we're beginning to determine from sequence analysis. Now, we have been focused initially on obtaining the sequences of individual species, and obtaining the sequence of a given strain of a species has been a major accomplishment. Now we have probably close to 1,000 species of microorganisms fully sequenced. What we are finding is that as we obtain more species for which there are many more than, let's say, 9 isolates sequenced, that's a very small portion of that 1,000. And we're finding some very interesting things as we sequence more strains from a given species. These data are from Welch et al. On E. coli, the non-pathogenic, this outer circle, the uropathogenic, and the enterohemorrhagic. And you find that only about 40% of the genes of that species are shared. and that there are genes that are specific to the non-pathogens, specific to the uropathogenic, and specific to the enterohemorrhagic. In fact, the more strains of a species that we sequence, we find that none are identical base pair by base pair, at least as yet analyzed. Now, let me see. Now, there are several comparative studies that have been done of multiple isolates. And this, I think, is very instructive because in some cases, about 25% of the genes were unique to a single strain. These are the strains that, the species that have multiple isolates, but I should add to that Bacillus anthracis, Vibrio cholerae, which I'll discuss in greater detail tonight, tularemia isolates, and we're now finding in the case of the tularemia bacterium that almost 50 strains have been isolated and none are absolutely identical. I'm having a little problem with this. Now, as we define a bacterial species, the core set of genes that are present in all the strains is around 80%. But as the number of genomes, that is, the number of strains are sequenced, we find that fewer and fewer genes are common to all of them. So this is a very interesting situation with respect to microorganisms. The number of genomes and the fewer new genes that we find, and we find that we reach an asymptote with respect to the central set of genes that identify a given species. So I like the definition that Claire Fraser-Liggett and her team, when she was at Tiger, now she's at the University of Maryland Medical School with her own institute. But the hypothesis is that there is, in fact, for an isolated lifestyle, limited access to a global gene pool, that there are core genes shared by all of the strains of a species, and then the non-essential genes that can be variable, laterally transferred. But for the open pangenome, that is a bacterium like Vibrio cholerae, which can be found in many different aquatic environments, there are the core genes, the non-essential, meaning genes that can be laterally transferred, can be deleted, can be acquired, and then genes that are strain specific, that are picked up by given strains of a species. I think this gives us a better understanding of what we have intuitively understood, those of us who have been using phonetic analyses for all of our career. Now, obviously, the ability to laterally transfer genes amongst bacteria, archaea, and even the eukarya, that is, share genes with the host eukaryote, is turning out to be much more frequently occurring than we had hypothesized previously. Now, trying to define a species based on size doesn't work very well because the very largest bacteria, the Thiomargarita namibiensis, is larger, in fact, than the human erythrocyte. And yet, the largest virus, the mimivirus, is actually larger than one of the bacterial species, the mycoplasma. So this has turned out, again, to be another area of great interest in the microbial world. The largest bacterium to date, probably will be found to be even bigger form subsequently discovered, is the Thiomargarita namibiensis, which has a huge number of sulfur granules within the cell. The smallest bacteria, the nanoarchaeum, are shown here that are probably about 2/10 or less of a micron, which means that we're reaching a smaller and smaller size in defining bacterial species. The bacterium Pelagibacter that was described by Steve Giovannoni essentially has a greatly stripped down DNA and is one of the smallest bacteria. This is found in the Sargasso Sea. It's about 100 nanometers in length. So if you lay out the bacteria according to the size, we have this is the overlapping genome size in members of the bacteria, which are in blue, the eukarya, which are in green, and viruses, which are yellow. So size no longer allows us to describe bacterial or microbial species. Let me talk a little bit about the environment as the residence of a variety of microorganisms. I will discuss very briefly biocomplexity, the use of information technology, which is very important in handling the large amounts of data that we're now generating in microbiology, and then a bit of the new applications of nanotechnology, and use the marine sciences as the example. Now, I'm bringing coals to Newcastle by describing metagenomics, and let me not go into any detail except to say that it is the technique that some of us, Bill included, some 20 years ago, were able to isolate total DNA from given water samples and probe it with a few gene probes. But now we're able to sequence it and to be able to get the identification of microorganisms by the pattern that's provided in the DNA sequences that we derive from the extracted DNA from populations. And this allows us to do very rich population analyses of microorganisms in the natural environment. This is a fairly new technique. The first publication was really in 1991. Co-workers isolated metagenomes about 1995, and now we have enormous numbers of complete sequences and the release of the Sargasso Sea sequences that occurred in 2005 and many, many more in the last 3 or 4 years. So this now is a tool, really, of environmental studies that's environmental microbiology that's proved extremely rich. Now, I like to use the data that Ed DeLong and his colleagues have published because it describes, I think, very nicely the mixed populations you have in an environment. And that is by studying the bacteria that are isolated or characterized at 40 feet, 200 feet, 400 feet in water samples in the sea. The unique DNA to those microorganisms at 40 feet is only about 20%. At 200 feet, about 35%, and roughly about the same at 13,000 feet. So only a portion of the DNA sequence is unique to the populations at each of these given depths. But what's interesting is that the population sizes in the euphotic zone are very large and much smaller until you get at the sediment-water interface in the ocean. And this would hold for rivers, streams, lakes, and probably even soil, though I haven't any data for soil. Now, the unique sequences, just as for the individual species, we find that the unique sequences for the given depths are really very few, that there is a great deal of shared species from the different depths, shared sequences of the species that comprise the populations at the given depths. I think this is very illuminating because it's describing in such detail now the populations of microorganisms that maybe 20 or 30 years ago were simply black boxes, the microorganisms. Now we find that they're enormously diverse, and populations are quite specific for given depths. There are also bacteria associated with specific components of a given environment. The bacteria that are found associated with protozoa, free-living bacteria, the planktonic forms of bacteria, bacteria associated with phytoplankton, and also with zooplankton and with the so-called marine snow, which is a mass of particulates in seawater. Similarly, we would find this in freshwater lakes and streams. Now, Ed DeLong speaks of systems biology, that is the composition of the bacterial cells, the viruses, bacteria, protozoa, that comprise the microbial populations that again are part of the larger communities and describe the full ecosystems. I think it would be helpful to also take the viewpoint that it is really biocomplexity that we now are putting our attention to. And that is understanding a given ecosystem from the molecular, the atomic, to the molecular, the cellular, tissue, organ, organismic, the habitat, the population, the community, ecosystem. And you may think that it's a little weird to talk about planetary and cosmos. However, if there is in fact life on Mars, then we are talking about microbial populations of other planets. And certainly in the cosmic dust, there does seem to appear the possibility that there is some sort of intergalactic microbial populations, perhaps, not Star Wars, but perhaps. Now, let me step back a bit and look at the populations of microorganisms that are found in, let's say, the marine environment. This is the context against which I want to build the case that many of the populations of pathogens and many of the strains of pathogens are really derived from the animals, the plants, and the environment itself. Here is Physalis, otherwise known as the man of war, which has a kind of a sail, and then very long tentacles to which are attached many microorganisms, and comprised of which are many microbial populations. In fact, if you want to look at the greatest diversity of all, it's on the Great Coral Reef, the coral reef barriers in Australia, the reefs of many of the tropical parts of the world. I think that if you are able to transfer to sound the great spawn that occurs twice a year on the Great Barrier Reef, it would probably be like Beethoven's symphony. This is a time when the diversity of the populations is expressed in the developmental stages of it. Now, some of the applications from the environment that are less known, from the marine biotechnology, which is now developing fairly rapidly. This is the nautilus shell. It has a very hard surface. In fact, this is a simulation constructed by chemists and engineers, and it has the tensile strength of the shell itself in the natural environment. And this is now being used perhaps even for airplane outer structure and for constructing devices that require a very hard external surface. Another aspect of the environment that's fascinating is that oysters, which settle on surfaces and create this strand of attachment, studies that we did some 20 years ago showed that the signaling was the bacterial film that formed on a surface, and that it was the L-dopamine produced by the bacteria that triggered the metamorphosing larvae of the oyster to settle and then to continue metamorphosis to the adult. Of course, L-dopamine, as you know, is used in treating Parkinson's disease. In the abalone, it's a different neurotransmitter, but these are produced by microorganisms in the environment. And in the case of the oysters, these compounds are a fundamental component of their life cycle. Sponges are also fascinating creatures. This is taken from courtesy of Discovery Channel, describing the pumping mechanism of the organism. Now, what's fascinating is when the genomics of sponges and other sea animals are characterized, we find that they're not all that different from us humans. In fact, even the larval stages resemble in some ways the developing human embryo. What's fascinating about understanding the marine context is that the Genome Center in Tokyo is run by a director who has taken all of the genes for the primitive forms to the most complex found in the marine environment, and has been able to, through the genome structure, just the genome sequence, follow the evolution of the human nervous system and also the human heart. And this comes from simply aligning the genes that have been isolated from the very primitive marine organisms to the more complex. So we are derived from the sea, and we can genomically even trace our development from the sea. There are other applications that are very important, and that is the giant squid axon is one of the most useful tools in neurobiology in understanding synapses and the function of the potassium and sodium channels in biology. And certainly the diversity of the zooplankton forms and the phytoplankton in their structures are really very dramatic. Again, some data from Ed DeLong and his colleagues, he is now at MIT, shows that there is a very specific adaptation of the rhodopsin-like compounds found in the microorganisms that are collected off the Monterey Bay area that are highly specific to the penetration of the wavelength of light to that given depth. And so this kind of evolution has been very dramatically demonstrated. Now, some of the work that's being done to monitor in the ocean, and this holds also for NEON, which is the ecological observatory, national ecological observatory sites that are being constructed on land. John Delaney and his colleagues have established what is called Neptune, and that is to establish sensors that allow us to measure the parameters, physical, chemical, as well as biological, across an entire ocean span, and then to transmit these data to satellites and then to collect it and to integrate it. Some of the interesting aspects of the deep sea microorganisms, in the case that I will describe in greater detail tonight, is that we discovered that the Vibrio cholerae bacterium has as its host the copepod, a zooplankton. And in a dive on Alvin, I noticed that there were what appeared to be copepods swimming in the periphery of the hydrothermal smokers and vents. And we collected some of these copepods from the deep sea vent. This is at a couple miles down, 250 miles off the coast of Oregon. And we found a very interesting surprise. Now this is a composite of one of the vents. This is by John Delaney in the area that we carried out the studies. Now, aside from the fact that within the vent fumes, there are the thermophiles, the hyperthermophiles. I think now it's up to about 140 or 50 degrees that they can be grown. Maybe it's 100, even higher. It might be 160. But in any case, the temperature, maximum temperature keeps going up. For us, well, let me go on to the next slide. This is one of the hydrothermal vent worms, that the Pompeii worm that has been genetically characterized, the gene sequence. And you all are doing a lot of work in the Yellowstone Park area. Let me go on to the next slide. For our samples that we collected at 2,500 meters in the East Pacific Rise, using the Alvin, we were able to compare the sequence that we had published in the year 2000 for Vibrio cholerae, confirming firstly that indeed there were 2 chromosomes. Up until then it had been hypothesized, mainly by mapping, the work of Jim Caper, former student, that there were 2 chromosomes. But until that point, it had been assumed that each bacterium had a single chromosome. Nevertheless, in 2000, John Heidelberg, another former student, then at Tiger, working with us at Maryland, and John Mekalanos at Harvard, were able to sequence fully the small chromosome and the large chromosome. So let me get to the punchline here, and that is the Vibrios that we isolated from the deep sea copepods were practically identical to the Vibrio cholerae. What we had done was simply to plate the crushed copepods on TCBS, the thiosulfate bile salts agar specific for Vibrio cholerae, and we got the bright yellow colonies, and we tested them phonetically, and then through some gene probes confirmed that they were Vibrio cholera-like. And then through the full sequencing, which we're now just finishing in collaboration with Los Alamos National Laboratory, Tom Breton and his team there, we were able to show that both comparison of the vent Vibrio with chromosome 1 and chromosome 2, that the blue are the amino acid sequences and the green are the nucleic acid sequences. But interestingly, when we compare with Vibrio vulnificus, another human pathogen, and a very nasty one, this particular bacterium is also showing very high similarity with the vent Vibrio, both the chromosome 1 and 2, and then finally Vibrio perihemolyticus. So we have perhaps what we might call, I don't say archaea, that has different connotation, but perhaps an ancestral Vibrio cholerae from the environment. So we're just now analyzing the genomes, and I'll show some more of the more complicated data tonight. We've been able to sequence about 27 Vibrio cholerae and a couple of mimicus, and we're now analyzing the biogeography of these fully sequenced Vibrio cholerae with respect to source of isolation, whether the Sudan in Africa or South America or Chesapeake Bay or Asia, and get a sense of the evolution of the bacterium. Now, another aspect of studying the environment, here's some data from, or at least a picture from Mark Wells at the University of Maine, who has formed some liposome devices that allows seeding, let's say an algal bloom or an oil pollution site, and being able to determine genetic responses to those given pollutants. So this is another aspect of environmental studies that are now possible. It would be important to emphasize as well the signaling capacity, which was really determined using marine systems. In this case, this is from Bonnie Bassler, now at Princeton University, and Ned Ruby and Margaret McFall-Nagay at the University of Hawaii, who showed that in the squid, the bacteria, the luminescent bacteria, when in sufficient numbers, light up and then diffuse the outline of the animal. It's perfect camouflage for the host. And the signaling molecules now are shown to exist, as you well know, in a large number of human pathogens and other bacteria. And that this is a mechanism that perhaps may be a means for interfering with or treating perhaps the multiply drug-resistant strains if we can find a way to interfere with the signaling mechanism. Because the signaling mechanism is critical in establishing the numbers of bacteria that allow then triggering the production of biofilms, which are a major mechanism in the disease process for many organisms. So understanding the signaling process and understanding the marine environmental origin gives us, I think, some insight in understanding human pathogens. Well, the diversity of the oceans, at least for the moment and for a while, is huge. And it's very, very important to understand, I think, the need for biodiverse environments in order to understand the health of environments. But it's also important to understand the interactions of microorganisms in the environment because it gives us, again, some insight into potential treatment against bacterial pathogens. In this case, this is work done by the team at the University of New South Wales, and they have shown in field trials that there are furanones produced by the microorganisms that coat surfaces that prevent biofouling. And you may ask, well, what's this got to do with medicine? Well, in fact, treating mice that are models for cystic fibrosis, it turns out that the furanones are very, very effective. This is the saline treatment of the mice, and these are treated with furanone. So this seems to be very effective against the gram-negatives that coat the lungs of the mice. So again, some information from the marine environment that gives us some insight on the interactions of microorganisms and on those characteristics that we consider to be pathogenic traits, which are really, let us say, ecological functions of the organisms in the natural environment. In fact, the cone shells, these organisms are essentially the pharmacists of the oceans because they produce a great variety of toxins, some of which are being considered as useful in alleviating pain in human diseases and human situations. And then, of course, there are the toxins that are powerfully pathogenic for humans, as occurred in a few years ago in this massive, this massive algal bloom this is a very different technological device. Sorry about that. Let me go back. You're getting my punchline ahead of time. Okay. A massive bloom that went from Maine all the way down to Buzzards Bay. And this essentially eliminated the harvesting of shellfish and also even, I think, some of the crustaceans in that area because of the toxins that were produced by the organisms in this massive algal bloom. Now, it's important to point out that in the environment, the marine environment particularly, a number of pharmaceuticals, the antiviral drug derives from marine sponges, the acyclovir. Another similar compound is an anti-cancer drug that has already gone to clinical trial. And then there are a number of molecular probes that have been derived from jellyfish, marine sponges, enzymes, certainly polymerases. There are new vent polymerases that are now in production at New England Biolab from marine sources, and then supplements and even cosmetic additives. from gorgonians, red algae, marine microalgae. The point is that I think we have to understand the environment as the residence of these organisms producing these interesting compounds that have applications in medicine but have environmental functions. And if we understand the environmental function, we can do a much better job in treating the diseases that are caused by the bacteria that they can be used to treat. And so the fundamental knowledge is still coming in, and commercialization of biomedical products and commercial applications I think are still sort of in the early stages, and marine biotechnology still has a long way to go before it reaches the applications of, let's say, medical biotechnology. Nevertheless, it would be a tragedy if we destroy that which we have not even named, studied, or even understood. And that seems to be the problem with the loss of diversity that seems to be occurring. The fact that codfish are commercially extinct, that's the term that was used by the Boston Globe about, I'd say 10 years ago. Haddock is getting close to the same situation. And the overfishing has reached a stage where I've heard on NPR just the other day that be prepared for $35 a pound salmon because of the crashing of the salmon stocks, at least in the Pacific Northwest. The diversity, I think, of the marine environment is probably the most aesthetic, and it's probably the greatest warning that we have, that if we lose the diversity, we lose the understanding of our of our environment and the functions thereof. We've really just begun to explore the full Fathom 5 and beyond. Let me close by saying that what has this got to do, a little bit about what this has to do with infectious diseases and the environment. It turns out that increasingly the so-called emerging infectious diseases are really resident in the environment, and it turns out that the seasonality of many infectious diseases are directly related to the environment. The best example is cholera, which I'll go into greater detail tonight. But even influenza, which has its seasonal peak in the winter months, and we have ascribed that to the fact that we're closer together, we're hovered around our stoves or heating units, and that's because we have greater transmissibility. Not so. It's just been shown, published in the last several months, that the influenza virus is far more infective and far more transmissible at lower temperatures. And so that accounts for the greater transmission, the greater infectivity in the winter months. It turns out that emerging diseases like the hantavirus in the Three Corners area, close to you all, broke out in major epidemic in 91, 92, 93. It was a period of the massive El Nino. There was a lot more rainfall. There was a whole lot more food for the deer mice. The populations increased. Transmission of the viruses amongst the mice increased, so the number of infected mice went up. And then the subsequent year, there was less food. The mice went into the barns and the homes to forage. They excrete the virus in the urine. The urine dries. Humans go into the barns, go into the basements, sweep up or kick up the dust, inhale, and then come down with a pulmonary disease. This is directly related to the environment. Of course, what's interesting is that Terry Yates and Gregory Glass, the late Terry Yates and Gregory Glass, developed a computer model to show where more likely the hantavirus would be found. And interestingly, it was in the area that was called the Canyon del Muerte by the Native American Indians, the Canyon of Death. They knew if they went into that area, they'd probably get sick and die, hantavirus. And we now know that since 91, 92, at which time, that time there was one or 2 virus strains. Now we have isolated, or scientists have isolated, maybe 50 or 60 strains of hantavirus across America, North and South America. So the so-called emerging viruses, the emerging bacteria, the emerging infectious diseases, we find have hosts and reservoirs in the natural environment. And it's important to understand the diversity, the complexity of the interactions, and the purpose of the toxins, the purpose of the signaling molecules, the purpose of the formation of biofilms in the survival and the metabolism of these microorganisms in the natural environment. So it's really biocomplexity, I think, that's the science of the 21st century. I hope I've provided some elucidation of what you may not have already known, and if I've only given you maybe a little bit more colorfully what you already knew, then I ask your forgiveness. In any case, that's essentially my message for this afternoon. I'll be happy to answer any questions. Thank you. Any questions or comments or expansion of what I've presented? Yes.
I like that you mentioned global warming this afternoon. You said you were going to consider that in the evening lecture. And I wonder if you could at least say a few words
Sure.
about that.
I think that it's becoming pretty well documented that infectious diseases have a broader spread and a wider range. Malaria is moving further northward and toward the southern pole area as well. It's also diseases like cholera are becoming more intense. The epidemics in Senegal, for example, right now the mortality rate is 20%, and it's usually 2 to 5% in even Bangladesh. And so there seems to be something going on. Fortunately, we've been able to get our hands on some of the strains, and we just received them last week. So we're in the process of sequencing them. We're going to first just run them through 454 and see if we find anything unusual, maybe multiple copies of the CTX gene or something unusual. In the case of cholera, it is very directly related to the environment, being associated with zooplankton. In Bangladesh, for the last 1,000 years, there have been spring peaks and summer, fall, massive peaks of the disease. And that fits exactly with the blooms of the plankton. First, sea surface warms, phytoplankton become abundant because of the sunlight, warm temperature. And then the zooplankton become abundant because they graze on the phytoplankton. And then the cholera, which are associated with, like we have E. coli in our gut, they have vibrios on their gut and on their surface. And the vibrios become abundant. And in those parts of the world, like Bangladesh, where they drink the water without any purification at all, then the epidemics occur. The bacteria attach to the gut just as they do to the surfaces of the copepod. And in fact, some studies have shown that there are receptor sites that are similar in invertebrates to which animals to which the cholera bacteria are found, and the human gut, villi. So there again seems to be that we are an inadvertent host, but the epidemics are occurring earlier and more intense. So this is a direct climate relationship. The work that's being done with dengue, with yellow fever, with vector-borne diseases are definitely showing longer seasons and more intense seasons. We're finding that with changes in habitat, I'll discuss this evening, that we have Lyme disease because the deer are migrating into populated areas because we have, in the Northeast, we have deforested. And so the deer have become more numerous and have moved into human areas. And Maryland, the Eastern Shore, is now a hotbed of Lyme disease. It's a very serious problem even Craig Venter got it twice because he has a boat that he sails into the eastern shore. So and it's been shown by Stan Shetler, who works at the Smithsonian, that flowers in the Washington, D.C. area over the last 14 years now have been blooming 2 weeks earlier. So there are distinct changes. And one of the problems that I think we will see, along with longer seasons and more intense infections, is a decoupling of life cycles of host and microorganisms that may have some rather unusual effects that we don't yet recognize. Another climate effect. Any other? Yes?
Are the genomic structures of the Vibrio similar? Having 2 chromosomes?
Yes, it has 2 chromosomes, very similar to the Vibrio cholerae and also Perihemolyticus and Vulnificus. And we're just now analyzing the data to try to find out what are the core genes for those Vibrios, and then what are the essential genes and what are the species genes, to find out just what are the differences. But I think it's very instructive that we've isolated from the deep sea vent around the smokers these deep sea copepods carrying the same bacteria that we find in nearshore forms, and actually in lakes. You know from history that cholera followed the settlers who moved out into the west, and it was shown by Dr. Des Marchelier in Australia that way upriver, beyond the tidal effects, there were copepods, and indeed she could isolate Vibrio cholerae. I think, this is just hypothesis, but I think that as the seas receded in the primitive world, and as the glaciers scoured out and left lakes, that the Vibrios, this is pure speculation, are perhaps, left behind, so to speak, and that they are related to the marine, freshwater, deep sea organisms, that they may in fact be perhaps an ancient form of bacteria. Yes?
You know how long, or any estimate of how long it is since these different species of Vibrio diverged?
That's a good question, and I think the way we will determine that is that there are these deep sea animals which have Vibrios in their guts. On each side of the African, off the south of Africa, and I think that, and they know when those 2 species of deep sea animals diverged. And if we isolate the Vibrios from them and determine what the differences are, we may be able to give a timeline to the divergence. And so that's one of the future experiments that I would like to do. Yes?
Given the degree to which microorganisms associate with a lot of higher organisms, humans, ungulates, etc., it's realistic actually for people to try to understand health and physiology of animals and humans without a full understanding of the microbial complement.
We're going to have some wonderful surprises. I noticed the other day that the human metagenomics of the oral tract, the mouth, has just been published. David Relman says we're 95% microbial cells and the rest are tissue cells. It's just that our tissue cells are bigger. And we're going to, I believe that, and I've already done a few experiments to show that it's the case some 10 or 15 years ago, that our body fluids are not necessarily sterile. That like cosmic radiation, there's a small background, and that we carry a burden, small numbers of cells that act to keep our immune systems pumped up, so to speak. And as we do metagenomics of blood that is taken aseptically, we're in for some surprises.
The cryptic microbiome.
Yeah. And we already see the implication of chlamydia in atherosclerotic disease. And so I think we're going to find some very, very interesting surprises. I also think we're going to find that there's a great deal more gene exchange than we expected. And in fact, in the human genome, I think we're already finding that there are what appear to be ancient bacterial genes. So, or at least that's what has been.
And viral too, I think.
And viral as well. So I think we're going to find that we're a very interesting dynamic system, yes.
I just wondered, I wanted to see if you could put on your former NSF director hat for a second. And
Sure.
Given these interactions between microorganisms and their hosts, and also given this interaction between climate change coming on as a very high-profile human stressor, but as you mentioned, that climate change is interacting with forest fragmentation and overharvest and these things. It seems like another axis of biocomplexity becomes human stressors, with climate change being one aspect of it, but these other human stressors being other ones. And these are things that have been carried along with NSF funding, but there hasn't been big NSF directives, at least not in recent years, including the human stressor in the biocomplexity. And I wondered what your thoughts would be, whether this is going to be on NSF radar, to be sort of putting human stressors interacting with microorganisms and host change.
I think that will be the case. I do. It's interesting. The biocomplexity story was started with, I think, $20 million back in '99, and by the time I completed my term as director, it was up to $100 million, $80 to $100 million. I think it's the initiative it was to be 5 to 7 years, and I think it's been renamed but continues, as I understand it. And I think it will take on a new dimension, because understanding the human-climate interaction will definitely be on the radar, regardless of who the next administration the leader is. McCain and Obama and Clinton have all said that climate change is a serious problem, and they will address it. I think that with the change will come some new directions for the NSF. It'll come probably with a transition team that will sit and review, as every administration change has done for the last 100 years, and will begin to make recommendations as directions to go in. I've had lots of discussions with the Japanese who have built the Earth simulator. The problem is it doesn't simulate the Earth, really, because there's no biology in it, none whatsoever. It simulates climate, atmospheric conditions and weather patterns, but it doesn't include just the point that you're making, and that has to be done. And we will get time on the Earth simulator anytime we need. I've had that promise from the Prime Minister. So I think we will see. If we don't do it, certainly it will be done in Japan. Yes?
I know you're associated with a couple of companies. I'm kind of curious, do you sense that there's any sea change in the pharmaceutical industry, natural products industries, et cetera, that really are starting to perk up their ears and recognize that there's a lot out there that we still don't know anything about that has a massive potential? Is there any kind of net influence in the exploratory research that you're sensing in these commercial firms?
I think it's beginning because we have this dreadful disaster that is much more serious than any of us, that any of the companies are willing to admit, and that's multiple drug-resistant infections. This is a very, very serious situation. I personally lost a relative just 2 months ago and watched him die because there was nothing that could be done. He was resistant to everything that they threw at him. He had Staph aureus. So this turns out to be much more frequent, and therefore, fortunately with genomics, it's understanding these interactions of the organism at a much more fundamental level to begin to attack their functioning, their metabolism, their division, etc., to be able to, if not kill them, at least put a stasis to their multiplication. So I think that we will see a much more sophisticated pharmacology. It won't happen overnight because we still have drugs that can be manufactured and profit that can be made, but I think the change will occur.
That's another tangential question.
Sure.
Comparative genomics is becoming huge, that's clear.
Right.
The movement in genomics is being driven by these new sequencing technologies that require just massive amounts of annotation and computation. Do you think that we have adequate computational tools and it's just a matter of throughput, or is there really a lot of room for a renaissance in computation in regard to being able to do comparative genomics?
You're asking a perfect question because, in fact, there will be a workshop next October that will be focused on just bioinformatics. Because, first of all, we don't have enough bioinformaticists to address the need, and secondly, we don't have the software that can really pull a lot of correlations that we're not able to do. So I think that it will change. Also, I think that it's been amazing. I've just completed a major study with some colleagues, John Jacobchek and with John-Sik Chun, my former postdoc, who's now a full professor at Seoul National University, of the whole DNA sequencing panoply, the whole panoply of it. And it's amazing how rapidly the expense is coming down and the sequencing itself is being done. With 454 and now Celexa, we're able to get sequencing done without necessarily annotating through the tedious methods that have been used. So I would see probably in the next 2 years the $1,000 genome that they've been pursuing happening.
So who's hosting that conference?
This will be done through NIH, NSF, Department of Agriculture, Department of Energy, even some of the even DOD. Because this is a major need. Yes?
As a former botanist and plant biologist, I have two short questions.
Sure.
One would be, you mentioned the phytoplankton as being involved somehow. I assume they had a more passive role to play in some of these phenomena than the copepods that you showed with the streaming bacteria, microbes coming from the top. From the copepod pore.
Yeah.
And at the bottom of the sea, I suppose.
Well, you mean on the copepod? Because we have done scanning EM, and it's really in the gut and around the axilla where there is some.
So these venting copepods.
Yeah.
But although is the material that's coming out venting, or is that microbial?
Some of it, yeah. Some of it is. Yes, indeed. There are microorganisms that live in the very, very high temperatures. It's hypothesized by John Barris and Delaney and others that in fact there's a continuum that runs beneath the sea floor from which vents sort of pop up here and there, and that there is a common kind of population of microorganisms that may live at even those very, very high temperatures, or at least survive and be transmitted.
There could be a whole stew of microorganisms, including phytoplanktons, that are being spread this way.
Yeah, it's a very interesting hypothesis. Controversial, but that's what science is all about.
And I assume, I'm intrigued by the possibility that somehow or other the viruses could play a role, or at least this marine environment could have played a role in the distribution of viruses, but that seems much less likely.
Well, there are, Zillig and others have actually, initially, they were the first to show viruses are present in the hydrothermal vents. They're part of the community as well. So they're high temperature viruses as well. Yeah, it's very interesting. Any other questions? Well, I'll have more to tell you tonight. Thank you.